This is just basically introduction of what that I do and what I participate in this group, and that I study the organic crops grown in Nebraska. And I specifically emphasize those components in organic crops based on the phenolic-based antioxidants. And the crops that I'm interested in are the soybeans, the grains, the corn, and the high oil sunflower seeds. I'm going to turn the slide next. And this shows an outline of what I'd like to present today. What are phenolic compounds first and foremost? What are goals and our outcomes from this research? And then to show you an example of some of that research that we are doing, and the data results from that one study, and then ongoing studies that are continuing through these studies, and some of our final impacts. So what are the phenols and the plant-based antioxidants? In general information, phytochemicals are those that are widely distributed through the nature. They usually are found in plants, but not necessarily so. You can find them in fish, you can find them in butterflies, certain insects, that sort of thing too. But they're most notably known for their presence in plants. Now, when they are present in plants, they're present in very small amounts. We're talking parts per billion, parts per million. They really are so much smaller than our main components of food. But what's really interesting about these types of chemicals is that they're very chemically diverse compounds. And we have found that consumption of foods rich in these chemical diverse compounds, or what we call micronutrients at this stage, is that we have been shown that they have been lower links to risk for cancer, heart disease, arthritis, Alzheimer's, diabetes, and Parkinson's disease. Amongst of them, there's others, of course, to talk about here, but I just named a few. So what a phenol is? A phenol is anything that has this ring structure, a six-faceted ring structure with bonds conjugated throughout the ring structure, and an OH group sticking from it. It then can morph into a higher structure, again with some more acetic type compounds coming out of them. I wish I could have a pencil to show you these. But you can see from the example I've given you, still see the hydroxyl groups with the ring structures and some of the different types of stereoisomers that can arise from this. These are called phenolic acids, and that they contain all the COHO groups. And then we got the stilphenoids to the right. The stilphenoids have two phenolic rings, and the stilphenoid I put up here because it's called resveratrol. Resveratrol has been implemented in so many different types of health mechanisms and health care products that I didn't think we should go without talking about these just a little bit. Oops, going the wrong way. Sorry about that. And then on top here, we've got some other polyphenols that group by, coming together more closely. They have some interesting qualities that most people don't study too often, but I do want to tell everybody that they still are in the plant system, and we don't know how they act synergistically with other plants to elicit a higher health benefit than what we would see if they were gone. And then we got my favorite, the flavonoids. The flavonoids are characterized by these three-winged structures, cyclic structures. You've got two close by each other that are bonded to one another, and another one that has a long chain and then is bonded further away. The one closest to the middle one is called A. The one further away from the middle is called B, and the one in the middle is called C. So when we test all of these compounds, we have a method in place that we can test all of them, and we call those total phenols, the total phenol test. When we go to flavonoids, we can further disintegrate them, deconstruct them into their individual structures. So here we got an isoflavone that goes into isoflavone, that goes into genistein, that goes into adenosine, by just virtues of little changes in these functional groups. Now, isoflavonoid is known for being in soy. You have, if you go down to the Phenols group, they are also shown by the fact that they have these double bond O group, but then it goes over to quercetin, and quercetin is a ubiquitous type of flavonoid that's found in most foods. So there's quite a few of these types of flavonoids, plus they can be conjugated to so many different types of sugars and glyco components, things like that, that we can get up to 4,000 to 8,000 different types of phenols. So to monitor all of these, we call them the total flavonoids. So what we want to do is that antioxidants or phenols are called secondary metabolites synthesized by different plants in response to stress. That is what they do. They try to defend the plant from any type of wounding, stage of drought, weed, insect pressure, UV radiation, soil nutrient content, location, infection, and cultivar. They are not considered the main component of foods at all. They are contained within the food, again, at very small levels, but the small amount present gives a lot for their pack. So what we wanted to do was determine the phenol flavonoid levels of organic grown groups in response to different factors, considering that they would be going through these different types of stresses all the time. And so our outcome was to provide information on the optimal crops, cultivars, and organic farming practices to implement in different areas throughout Nebraska that promote some healthy crops that are prevalent in our state. Here's a picture of Nebraska with the various organic lands that have been set aside by the university for us to do these types of experimentation. Charles has talked about Haskell Lab, which is shown at the northeastern side of the state. Then we have this Ag Research Development Center, which is Meade, and then we have the Central Ag Lab Clay Center, which is about the middle south central part of the state, and then over by Sydney, the High Plains Ag Lab. And we have organic plates or organic plots in each of these areas now. So one of our first studies is that we wanted to determine how phenols and flavonoids could be affected when grown through these different areas in in our state, but in regions that are very different in terms of their agricultural, you know. So we had 19 different wheat lines grown under organic conditions. We grew them in 4 locations throughout Nebraska, and then we monitored for their total phenol flavonoid levels, and then we monitored them for their antioxidant capacity. We are in the process of doing peroxamates, which includes protein, carbohydrates, water, minerals, let's see, fat, and carbohydrates there too. And our long-term related back to yield, and we want to relate this back to yield and other quality characteristics that our colleagues are also working on. So just to show you a couple things, because we have so much going on, I wanted to show you the differences in total phenols in terms of what happens when they were planted in Haskell Lab, Ag Lab, the Ag Research Development Center in Meade, and South Central Ag Lab and Clay Center. And if you look at the top of Haskell Lab, you can see that there are differences in the amount of phenols being produced in response to the different lines of wheat that were used. The most predominant phenol was by millennium. The least amount of phenols was overland. However, what I thought was interesting about Haskell Lab is that even though you saw these high and lows, the overall variability between each of these lines were very low, which, you know, very low variability is a good thing. And our average between all of those types of lines were 0.389 mg per gram. That's not the highest we've ever gotten from Haskell. We have gotten up from Haskell to be about 0.5, 0.6 grams per mg. Milligrams per gram. That was in 2008. So, you know, but we had a lot more variability then too. So what we have to determine is variability worse or getting higher yields with higher variability a better option to go to. For the Ag Research Development Center, we found that the highest yields were NEO 5425. Again, it wasn't that much higher, but we got about 0.4 mg per gram for this phenol. But the rest remained about fairly the same, not a lot of variability between them, and together as a group we obtained about 0.382 mg per gram of phenols. And for the last one, clay counter, this was the lowest in terms of producing phenolics. It did show several trends, and that a lot of the lines show various trends, with the highest trend being overland, with the lowest trend being buckseed. But again, I was happy to see that the variability was more consistent than what we have seen in the past, even though in the past we have gotten to be about 0.6. So, but that it could go back and forth as well. Now for the flavonoids, and flavonoids, because they are known through health promoting research that flavonoids are probably a little bit more potent than the flavonoic acids, things like that, the other things that I've shown you, even though they are around at a lower amount. So for the Haskell Lab, there was no statistical difference between any of those lines during the wheat lines during 2011. they all showed about 0.1 plus or minus 0.02. For their overall amounts to be about, average amount to be 0.099. We did see a little bit more higher averages with the mead, and that was 0.166. But again, the variability, although it doesn't look like it compared to the Haskell, was actually a lot less than what we saw in 2008. But the yields were about the same. The yields don't change with the flavonoids too much throughout the years. So, and we see about the same McGill, NE05, all of those being the higher producers. Going back now to Clay County, again, you see not a lot of variability between the flavonoids. Some of it, the NE093 show high yields, the Darrell shows lower yields. For overall average of 0.0988, so not much different than Haskell labs. And if you look to the right too, I have the average antioxidant capacities, and they were about 440 micromole per 100 grams of product, compared to the Haskell, which was 4,008 micromole per 100 grams, which was a lot higher than what we saw with the mead, even though it seemed that they might have a little bit more phenols there. And after looking at all of this data, I am really getting more and more convinced it's just not total that we're looking at. And the total thing that we're looking at is that what we need to do more compositional analysis of each of these to determine what types of flavonoids or phenolic acids are really exerting their effect. So we do have data from 2008 to 2011 to look at that type of data, and we are going through each of them methodically to really understand the genetics versus the environmental have on each of these wheat lines at the different types of the different locations in Nebraska. But other work that we are doing that I don't want to count out, and because of lack of time, is that we are evaluating organic grown crops for phenolic compounds and their health promoting their health promoting benefits, I should say, across cross types. We do know that sunflower seeds produce much higher phenolic and flavonoids and antioxidant capacity than wheat does. And we do know that soy does through certain time too. We just don't know how they are affected by cultivars, by locations, by nutrient treatment across years, disease stressors. And when I say disease stressors, I'm not only talking about the diseases that the crop itself can face, but I'm also talking about the disease stressors that humans eat, and we also can assimilate into our own bodies. And we also have not done any work with conventional crops. We're talking to a few people about looking into this, but this is really a major area that we need to address to understand conventional crop farming practices with organic crop farming practices and how they all help produce a better, healthier food. And so the final long-term impact of my research, which I hope will integrate to everybody else's research, is to produce a healthy crop, be it from what we eat, but how we grow them and how it affects the environment and so forth.